Hello dear students, I'm your instructor Abidjan. This is lecture number 21 that will cover the basics of DNA replication from the course of molecular biology. Learning objectives. By the end of this section, you'll be able to do the following. Explain how the structure of DNA reveals the replication process. Describe the Messelson and Stahl experiments. What was clear and what was not clear. The elucidation of the structure of the double helix provided a hint as to how DNA divides and makes copies of itself. In their 1953 paper, Watson and Crick penned an incredible understatement. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanisms for the genetic material. With specific base pairs, the sequence of one DNA strand can be predicted from its complement. The double helix model suggests that the two strands of the double helix separate during replication, and each strand serves as a template from which the new complementary strand is copied. What was not clear was how the replication took place. There were three models suggested: conservative, semi-conservative, and dispersive. The three suggested models of DNA replication. Gray color indicates the original DNA strands, and blue indicates newly synthesized DNA. In conservative replication, the parental DNA remains together and the newly formed daughter strands are together. The semi-conservative method suggests that each of the two parental strands acts as a template for new DNA to be synthesized. After replication, each double-stranded DNA includes one parental strand, or old strand, and one new strand. In dispersive model, both copies of DNA have double-stranded segments of parental and newly synthesized DNA interspersed. Understanding how DNA replicates. Messelson and Stahl were interested in understanding how DNA replicates. They grew E. coli for several generations in a medium containing a heavy isotope of nitrogen 15N, which gets incorporated into nitrogenous bases and eventually into the DNA. Messelson and Stahl experimented with E. coli grown first in heavy nitrogen 15N, then in 14N. DNA grown in 15N is heavier than DNA grown in 14N. This is 15N. And this is 14N. Being heavier, the 15N DNA sediments to a lower level in cesium chloride solution in an ultracentrifuge. When DNA grown in 15N is switched to medium containing 14N, after one round of cell division, the DNA sediments halfway between 15N and 14N levels. Indicating that it now contains 50% 14N. In subsequent cell divisions, an increasing amount of DNA contains 14N only. These data support that semi-conservative replication model is more promising. The E. coli culture was then placed into medium containing 14N and allowed to grow for several generations. After each of the first few generations, the cells were harvested and the DNA was isolated, then centrifuged at high speeds in an ultracentrifuge. During the centrifugation, the DNA was loaded into a gradient and spun at high speeds of 50,000 or 60,000 RPM. Under these circumstances, the DNA will form a band according to its buoyant density, the density within the gradient at which it floats. DNA grown in 15N will form a band at a higher density position, that is, farther down the centrifuge tube. Then grown in 14N. Messelson and Stahl noted that after one generation of growth in 14N, after they had been shifted from 15N, the single band observed was intermediate in position in between DNA of cells grown exclusively in 15N and 14N. This suggested either a semi-conservative or dispersive mode of replication. The DNA harvested from cells grown for 2 generations in 14N formed two bands. One DNA band was at the intermediate position between 15N and 14N, and the other corresponded to the band of 14N DNA. These results could only be explained if DNA replicates in a semi-conservative manner, and for this reason, therefore, the other two models were ruled out. During DNA replication, each of the two strands that make up the double helix serves as a template from which new strands are copied. The new strands will be complementary to the parental or old strands. When two daughter DNA copies are formed, they have the same sequence and are divided equally into the two daughter cells. Section summary. During cell division, each daughter cell receives a copy of each molecule of DNA by a process known as DNA replication. The single chromosome of a prokaryote or each chromosome of a eukaryote consists of a single continuous double helix. The model for DNA replication suggests that the two strands of double helix separate during replication, and each strand serves as a template from which the new complementary strand is copied. In the conservative model of replication, the parental DNA is conserved and the daughter DNA is newly synthesized. The semi-conservative model suggests that each of the two parental DNA strands acts as a template for new DNA to be synthesized. After replication, each double-stranded DNA retains the parental or old strand and one new strand. The dispersive model suggested that the two copies of the DNA would have segments of parental DNA and newly synthesized DNA. The Michelson and Stahl experiment supported the semi-conservative model of replication, in which an entire replicated chromosome consists of one parental strand and one newly synthesized strand of DNA. Critical thinking questions. How did scientific community learn that DNA replication takes place in a semi-conservative fashion? My answer to this question will be that it is actually Michelson's experiments with E. coli grown in 15N, which is the heavy isotope of nitrogen, they deduced this finding. The second question is that if we imagine that the Michelson and Stahl experiments had supported conservative replication instead of semi-conservative replication, what results would you predict to observe after 2 rounds of replication? Now, imagine the Michelson and Stahl experiments had supported conservative replication instead of semi-conservative replication. So following 2 rounds of conservative replication, 2 bands would be detected after ultracentrifugation. A lower or heavier band would be at the 15N density and would comprise 25% of the total DNA. A second higher or lighter band would be at the 14N density and therefore would contain 75% of the total DNA. Critical thinking questions. Who were the first to suggest that one strand of the DNA might act as a template for the synthesis of its complementary strand? In Watson and Crick's paper on the model of DNA double helix, they ended with a statement that it had not escaped their notice that the specific pairing they had postulated immediately suggested that one strand might be the template for the complementary strand synthesis. Who for the first time demonstrated the semi-conservative nature of replication of eukaryotic chromosome? Semi-conservative nature of replication was first demonstrated by J. Herbert Taylor, Philip Wood, and Walter Hughes on root tip cells of broad bean, which is the common name for Vicia faba. Why the base pairs have opposite polarity in the DNA double helix? The two strands having complementary base pairing have the same helical geometry but have opposite polarity. This is because the strands are held together and are anti-parallel in nature. Who showed from enzymological proof that DNA alone functions as the template for the synthesis of new DNA strands? Within 5 years of the discovery of the double helix, evidence emerged for the separation of the complementary strands during DNA replication, as suggested by Watson and Crick. This firm enzymological proof was first provided by the Michelson and Stahl's experiment of semi-conservative way of replication in 1958. Replication of DNA ends are carried out by which polymerase? Replication of DNA ends, that is, the telomeres, cannot be done using polymerases. Therefore, a cell has a unique enzyme, telomerase, which uses its own unique mechanism for telomerase replication. Replication does not take part in gene expression. Why? Replication is the copying of the gene in double, but plays no role in the production of protein, thus expression of the gene. Transcription is the production of RNA from DNA, and RNA processing is used for stabilizing the RNA in cytosol and excision of non-coding regions. Translation is the major process that leads to the formation of polypeptide chain. Could multiple copies of RNA be formed at the same time? Yes. The RNA produced does not remain base paired to the template DNA strand and is displaced only by a few nucleotides behind the transcription site. Thus, another RNA polymerase can attach itself to the DNA template, facilitating multiple RNA production. Amino acids, prior to their incorporation into polypeptide, must be attached to a special adapter molecule. Who proved this? In 1955, Francis H. Crick proposed that prior to the incorporation of amino acids in polypeptide chain, they must be attached to an adapter molecule that can interact with the 3 nucleotide long coding units of mRNA. This was finally proved in 1957 by Paul C. Zamechnik and Hoagland, and they demonstrated that amino acids were carried by the transfer RNA or tRNA to the ribosome for its transfer to the growing polypeptide chain. Why mutation in DNA is necessary for the purpose of evolution? As you know that mutations are actually DNA variations, and they may be of different types. These sequence variations actually facilitate the change of bases within a nucleic acid molecule. And if this change encodes for a viable amino acid, which in turn may lead to the synthesis of a different protein, this protein exerts a phenotypic character to the organism which may be different from the wild type character, or may generate a unique character itself, thus leading to evolution. What are plasmids? Plasmids are extra chromosomal circular molecules. They are actually found mostly in prokaryotic cells, and they are auto-replicating small circular double-stranded DNA molecules which generally carries certain genes for the advantage of bacteria, such as antibiotic resistance. What stores the genetic information in DNA? Genetic information is stored in the sequence of nitrogenous bases, as there are of four types, and they are adenine, thymine, guanine, and cytosine. The order of their occurrence decides the mRNA sequence, which in turn codes amino acids and leading to synthesis of proteins. Sugar and phosphates form the backbone and are common to all, whereas polymerase helps in replication of DNA template. Does DNA always carry the genetic information for all organisms? No. Generally, as you know, there are certain viruses in which RNA carries the genetic information. They are known as RNA viruses. For example, retroviruses, influenza viruses, coronavirus is also an RNA virus. What is the major type of RNA produced in the cell required for translation? Generally, ribosomal RNA is of four types: 28S, 5.8S, 18S, and 5S. These subunits comprise the 80% of the total amount of RNA produced in a cell and are majorly required for translation, as these ribosomal RNAs along with proteins form ribosomes, which reads the codes for translation. Which molecule is not abundantly found, though being of critical importance for biological mechanism? Lipids. Though lipids are of essential use in the cell for the formation of cell wall, it is generally found in association with either phosphates or polysaccharides. On the other hand, proteins, nucleic acids, and polysaccharides are all macromolecules and are of essential use for the cell. For example, proteins such as enzymes regulates biological functions. Nucleic acids carry genetic information, and polysaccharide function as either storage of energy or act as a structural polymers. Is a DNA double helix, the bases are held together by hydrogen bonds? What is the nature of these hydrogen bonds? Hydrogen bond is actually a weak non-covalent interaction. This occurs between two molecules resulting from an electrostatic attraction between a proton and an electronegative atom of the other molecule. No share of electron takes place, which is the criteria for non-covalent bonding. The bibliographic information. Stay curious and all the best for your life ahead.